Welcome to today's Explainer. We're diving right into how modern biotechnology has literally flipped the script on how we treat and detect diseases. For the longest time, human medicine was kind of like a game of educated guesses using some pretty crude tools. But today, well, we're operating straight at the molecular level, which fundamentally changes what it means to heal. We're going to look at the unbelievable strides science has made to boost healthcare outcomes across the board.
Now imagine this. What if you needed the pancreases of literally thousands of pigs and cows just to manage a condition like adult-onset diabetes? I know, it sounds straight out of science fiction, but historically, patients actually had to do exactly that. It sets a pretty jarring baseline for just how far our medical treatments have evolved. We used to have to look outside of ourselves, often relying on mass animal harvesting just to get the compounds our own bodies were failing to produce. And, as we're going to see, that approach came with some major risks and limitations.
So, here's our roadmap for today. We've got four main stops. First, the recombinant DNA revolution. Second, engineering human insulin. Third, gene therapy in action. And finally, the power of early detection.
Okay, let's jump straight into Section 1, the recombinant DNA revolution and engineering safer treatments.
You see, historically, those conventional pharmaceuticals derived from animals, they often triggered wildly unpredictable immune responses. Think about it. When you take a medication derived from an animal source, your body naturally recognizes those foreign proteins as invaders, and that can lead to some really dangerous allergic reactions. But today, the game changer is recombinant therapeutics. By leveraging recombinant DNA technology, we can actually engineer treatments that are incredibly safe and completely bypass those unwanted immune responses. The result is a vastly more predictable patient experience. In fact, right now, there are around 30 of these highly predictable, engineered recombinant drugs officially approved for use worldwide.
And what's genuinely amazing about this is the growing global access to these modern marvels. Out of those 30 life-saving recombinant drugs approved globally, 12 of them are currently available right now in India. It really goes to show how this once highly experimental biotechnology is rapidly becoming a standard, everyday pillar of global healthcare.
Moving right along to Section 2, engineering human insulin, which was an absolute molecular breakthrough.
If you contrast our old reliance on pig and cattle pancreases with today's genetically modified bacteria, you can immediately see why modern insulin is such a massive upgrade. Traditional animal sourcing meant there was this constant lingering risk of allergic reactions, because at the end of the day, it was still a foreign animal protein entering the human body. But genetically modified bacterial insulin, it completely rewrites the script. It streamlines production and guarantees a highly reliable, consistent, and safe supply, entirely without those old allergy risks.
Now, what's truly fascinating is exactly how scientists pulled this off. It turns out, human insulin's pretty straightforward on a molecular level. It's essentially just two polypeptide chains, simply known as chain A and chain B, and they're linked together by disulfide bonds. That's it. Because of this structural simplicity, scientists realized, hey, we can easily program bacteria, literally genetically modifying them to act as microscopic factories that produce these exact human components. It's brilliant.
But even with all this incredible molecular engineering, there's a quick caveat we have to remember. Whether it comes from an animal or a high-tech modified bacterium, insulin breaks down entirely in our digestive system. And that is exactly why oral administration is, unfortunately, totally ineffective. So, this life-saving medication still has to be injected, rather than just taken as a simple pill.
All right, Section 3, Gene Therapy in Action, where we start fixing our very own code.
We've actually evolved from just treating symptoms, like routinely injecting insulin, to directly rectifying the actual genetic defects that cause hereditary diseases. We can ground this incredible science in a really profound landmark moment, the year 1990. This was the very first successful clinical application of gene therapy. It was performed on a young girl suffering from adenosine deaminase, or ADA, deficiency. ADA is absolutely crucial for maintaining healthy immune functions, and without it, her immune system was practically non-existent.
Before 1990, the options for a disease like ADA deficiency were pretty bleak. Traditional treatments, things like bone marrow transplants or enzyme replacement therapies, were tried. But while they offered a little bit of temporary relief, they weren't completely effective. They were really just managing the fallout of the disease, not curing it. This highlighted a desperate need for a much more precise cellular intervention. And that's exactly where gene therapy stepped in, directly targeting and altering the underlying genetic defect itself.
You can think of this process as a microscopic rescue mission. So how does it actually work? Well, first, doctors extract lymphocytes, which are a specific type of white blood cell, directly from the patient's bloodstream. Next, they take a normal, healthy cDNA, which is basically a pristine working copy of the gene, and they introduce it into those extracted cells using a retroviral vector. Think of the retrovirus as a tiny delivery truck dropping off the correct genetic code. Finally, they return these newly modified, fully functional cells back into the patient so they can get to work fighting the deficiency.
However, we aren't exactly at the finish line just yet. Because these genetically altered cells do eventually die off naturally over time, the treatment isn't a permanent one-and-done solution. The modified cells just don't last forever. This means the patient still requires periodic infusions of these altered cells to keep their immune system functioning. So it's a profound fix, for sure, but still a temporary one.
Now for our final stop, Section 4, the power of early detection, or seeing the unseen.
As we all know, the most effective disease management absolutely hinges on detecting the threat as early as possible. Traditional methods, like your basic serum or urine tests, basically force us to wait. They really struggle to detect diseases early, which means by the time a test finally comes back positive, the disease has usually already progressed and the symptoms are fully visible. But modern molecular diagnostic tools? They act as our ultimate early warning system. They let us see the unseen, spotting diseases at the microscopic level long before you ever start feeling sick.
One of the absolute heavy hitters in this early detection arsenal is PCR, which stands for polymerase chain reaction. PCR basically acts like a molecular magnifying glass. It takes impossibly tiny amounts of nucleic acids and amplifies them. By multiplying these microscopic traces of a disease, doctors can easily spot what's lurking in the background. This technique is tremendously valuable, especially for identifying diseases like HIV, or for zeroing in on specific genetic mutations linked to cancer way before any clinical symptoms ever show up. Similarly, we have ELISA, or enzyme-linked immunosorbent assay. Now, rather than amplifying DNA like PCR does, ELISA takes a different route. It tracks down the unique biological footprints of an infection. It leverages antigen-antibody interactions to do this. So it can detect an infection either by spotting the actual antigens, which are the foreign substances from the pathogen itself, or by detecting the specific antibodies that our own immune system is producing to fight it off. It's incredibly clever.
So as we look back at these profound biotechnological strides today, from literally programming bacteria to manufacture human insulin, to amplifying invisible traces of cancer, to editing the very code of our immune cells, it leaves us with a truly awe-inspiring question. We know that current gene therapies still require periodic infusions. But ideally, as this science progresses, what happens when we can permanently correct a defective gene, like that ADA gene, right at the embryonic stage? A lasting, permanent fix that stops hereditary diseases before they even begin. That right there is the true frontier